Good morning everyone. Today in this video I'll be talking about uh, those scientists who contributed for soil microbiology. Okay, now soil microbiology means there is obviously microbes like bacteria, fungi, actinobacteria, etc. Okay, how these type of microbes are related with the soil, what are their impacts in the soil fertility or their impacts in biogeochemical cycles, all was discovered first by a scientist. He was a very eminent scientist named Martinus Beijerinck. Okay, Martinus Beijerinck. He was such an eminent scientist who first discovered three things. One, that is enrichment of culture media. Okay, now what is enrichment of culture media? Suppose you have only nutrient agar. Nutrient agar is not enriched media. Nutrient agar is all purpose media. It's a defined media or synthetic media which supports the growth of various kinds of microorganisms, different type of bacteria that can be inoculated, and they will show their growth on nutrient agar media. But if you want to specifically isolate nitrogen fixing bacteria only from a soil sample dilution, then what you will do? That was invented by Martinus Beijerinck. That is the enrichment of the culture media. Suppose if you want to isolate solely nitrogen fixing bacteria, how you will do that? First, you need a specific media that will support the growth of only nitrogen fixing bacteria from the soil in the lab, not the other bacteria. If other bacteria will also, if it is grown on the same media, then you cannot be able to differentiate that who are the nitrogen fixers or who are not the nitrogen fixers. So if you want to select the nitrogen fixing bacteria, isolate specifically from that typical kind of a bacteria from the soil, then you need a media that is basically minimal media. Okay, it's basically without nitrogen, it's a minimal media with all the constituents and a sugar or a carbon source. Only the nitrogen source will not be provided in the minimal media. So if the nitrogen fixing bacteria, they are present in the soil sample, and if you culture the soil sample for about 24 to 48 or 72 hours, maximum for 72 hours, nitrogen fixing bacteria, or even nitrogen fixing bacteria sometimes take more time, like 7 days, 14 days, it depends on the type of the bacteria. So they will definitely show their growth on the nitrogen fixing minimal media. So that is the selective media which basically select the growth of only nitrogen fixing bacteria, not sulfur producing bacteria or siderophore producing bacteria or phosphate solubilized bacteria. No, only nitrogen fixing bacteria. So that is the enrichment of the culture. So that is the first invention. Now the second invention, that was nitrogen fixation. He first invented the thing that some bacteria that is present in the rhizosphere of the soil, suppose this is a plant, okay, this is the root, These are the nodules of the legume plants. Okay, now there, so this is basically the rhizosphere, this portion. In the rhizosphere, there are bacteria who are able to make nodules. They will show their growth inside the nodules and fix atmospheric nitrogen in the plant in a way that the plant can utilize that fixed nitrogen for its own use, like ammonium. Because atmospheric nitrogen cannot be fixed by the plants itself on their own, they are not able. So only rhizospheric bacteria or the endophytic bacteria who are able to fix atmospheric nitrogen and convert it into the ammonium or something like that, then only the plant can take up those nitrogenous compounds and use for their own growth and development. So that is the nitrogen fixing bacteria. And the most important thing, this indicates the symbiosis process in the soil. So that is nitrogen fixing bacterial invention. That is the second point. And third one is sulfate reducing bacteria. Invention of sulfate reducing bacteria. Those bacteria who can reduce the sulfate, use that sulfate for their own use and also for the plants, and induce the soil fertility. That was the third invention. So there are 3 total inventions. Okay. With this, the contributions of Martinus Beijerinck in soil microbiology, that was ended up. There are more eminent scientists who have contributed for soil microbiology. I'll discuss about them in my next videos, and I'll upload a short on this eminent scientist as well. If you really like, please subscribe my channel. You can check these topics in my channel and share with your friends. Thank you so much.